Today I will give a talk about the work I did at the University of Amsterdam under the supervision of Associate Professor Dr. Jeroen Kool, and my title was From Necrosis to Hemorrhage, Snake Venom, Cytotoxicity, its underlying mechanisms, and the high-throughput methods to study it. So first, cytotoxicity, basically the definition is cytotoxicity to cells causing cell damage or cell death. And this leads to a variety of pathologies, including pain, necrosis, edema, blistering, rhabdomyolysis, hemorrhage. And some examples we can see here. Just quickly, there's a pointer. For example, this one is a bite by Boitropsia areata in Brazil, where we can see this very prominent blistering. Here is a bite from Callosolasma rhodostoma, I think from Thailand. And yeah, we can see some necrosis, blistering as well, and also some hematoma under the skin here. And this here, we see gum bleeding after the bite from Aedes ocellatus in Western Africa. So cytotoxicity is not that straightforward. We have different mechanisms that can cause cytotoxicity. First, we have the cellular membrane destabilization. Toxins that cause cytotoxicity this way are most prominently your three-finger toxins and your PLA2s, which belong to the major groups in snake venoms, and also beta-defensines. Then we have cytoskeleton modifiers, which are your C-type lectins and disintegrants. And then today we will focus on the extracellular matrix degradation, or for short ECM degradation. And this is caused by snake venom metalloproteases and hyaluronidases, with the SVMPs being of particular interest for us here. Here we have an example of a structure of a SVMP, in this case, borthropazine from Boitropsia areata venom. This is a P3 SVMP and a dimer, as you can see. So these are fairly large proteins. Okay, so then the ECM, which is the target of one of the targets of SVMPs, it is fairly complex in structure. The ECM basically separates into the interstitial matrix, which we can see above here, which mainly consists of collagens 1 and 3 and some collagen 12 and collagen 14. And then we have the basement membrane, which is mainly made up of collagen type 4, perlecan, and other proteoglycans like nitrogen and laminin, and these get cleaved by snake venom metalloproteases. Mainly the basement membrane components are attacked by these types of toxins, and different subclasses, which are your P1, P2, and P3 SVMPs, cleave different components or the same component at varying rates. So they have different cleavage efficiencies. Then the hyaluronidases, they exclusively cleave the hyaluronic acid, which we can find basically intermingled in the spare room, so to speak, between the collagen fibers, and acts like a spreading factor. What is important to note that SVMPs, they constitute by acting on different kinds of basement membranes, they cause different pathologies. So in muscles, it's muscle necrosis. In epidermal BM, it's blistering. Then when capillaries are affected, they cause hemorrhage by simply destroying the capillary wall, or destabilize the BM stromal interactions and release bioactive peptides from ECM by degrading said structure. Then we come to the species that we included in our study. We covered a broad spectrum regarding range and species, so from all over the world, starting with Boitropsia areata in Brazil, representing the Americas and the pit vipers. Then Aedes ocellatus of Western Africa, being a member of the true vipers. And then we have Callosolasma rhodostoma, which is another pit viper from Southeast Asia. Then we have 3 elapids. We selected Naya and Naya mosambica because they are known for being extremely cytotoxic, at least certain populations, and then Dendrosus polylepis, kind of like, yeah, so to speak, a negative control or a venom that is more known for its neurotoxic effects. Then we used an assay utilizing gelatin and collagen type 1 as substrates to study the cleavage rate of these different venoms. Here we have the elapids. First, we can see that the 2 Naya species are fairly similar in activity, with Naya mosambica being a bit more potent towards the gelatin, as well as the collagen type 1. What I have to note here, we required 4 times as high a concentration of the collagen to achieve our results that had a nice resolution and everything. So it's more difficultly cleaved, but we will come to that later. Then Dendrosus polylepis, despite having the highest share of SVMPs in its venom of the tested species with 3.2% versus only slightly lower 2.6 for Naya mosambica, and then around 1% for Naya, has the lowest activity. But nonetheless, it's not absent of any cleavage activity, especially at high venom concentrations. The substrates get cleaved relatively efficiently. In the scale here is in relation to a positive control, with this, which is a collagenase from a Clostridium bacterium. And as you can see, we have about 30% relative activity for the highest venom concentration here for Dendrosus polylepis. So it's not non-toxic to these components at all, but not as toxic as the other 2 elapids. Then we have the real, kind of like very toxic venoms that kind of drench through this, through these substrates like a freight train. They are very potent, even more potent than our collagenase as a positive control, with Wachtel areata being the most potent, and which is kind of interesting that this one actually has the lowest SVMP share in its venom from the 3 Viperidae species that we tested. After that, Callosolasma rhodostoma is the most potent, and Aedes ocellatus is the least potent, despite having over 70% of its venom made up of SVMPs. Again, here we can also see slight differences between the different substrates, but overall it continues the same picture that we have also with the elapids. This assay that we developed also allows us to study fractionated venoms. So in this case, we fractionated the venoms of the 3 very active vipers, and here we can see that both substrates, so collagen type 1 and gelatin, get cleaved by the same toxins at giving similar patterns. Here we use the same concentration, so for some reason the fractionated venoms also work quite well with a lower concentration of collagen type 1. Here we can see with Botryopsia areata, the activity peak correlates very nicely with this very prominent peak here, and also this minor peak here. And the active fractions elude between about 7 to 10 minutes. while for cholecystomas they elute a bit later, between 8.75 till about 11 minutes. But also here we have two nice peaks in our bioassay traces, which also correlate with these two nice peaks here in our size exclusion chromatogram. Then Tuerchis ocellatus, as expected, because it's pretty rich in SVMPs, as we saw before, they elute over a large period of time. So about 7.45 to about 11 minutes. And the bioassay traces also nicely correlate with respective peaks in the size exclusion chromatogram. Then this would allow for future studies to identify our respective active fractions, but we will go to that now in our conclusions and outlook. So first of all, viper venoms exhibit higher activities than leopard venoms, which is kind of expected because vipers are historically often characterized as being the cytotoxic snakes compared to the leopards, which are often more associated with neurotoxicity. But of course, with, for example, Naya and Naya mozambica, which are both highly cytotoxic, but these are then different mechanisms from the ECM degradation. So these we cannot detect with our assay. Then gelatin was cleaved more efficiently than collagen type 1. This is also expected because collagen type 1 is an actual component of our ECM, while gelatin is pre-digested collagen type 1. So it's easier for the proteases to cleave the gelatin, but it makes for a good, simple substrate for such an assay, and gives one an idea if venoms are actually active, and then further testing with actual components can be added afterwards if you want. Then, as we saw as well with the vipers and the leopards, that the venoms with the highest share of SVMPs did not show the highest activity. So we don't have like this nice correlation, the higher the SVMP content, the higher the activity. This is not how this works, because SVMPs, some SVMPs do not act on the ECM, they act more on the blood chemistry, and others are not as efficient at cleaving the ECM than others. Then it allows us to study the ECM degradation in vivo in high throughput. So we can do this with crude venoms or separated venoms. So we can identify active fractions after size exclusion. And then afterwards, the next goal would be then to identify the active toxins in these respective fractions to 100% confirm that SVMPs are at play. But from the literature, this is most likely to be expected. Then detection of underlying toxicities, potentially causing problems in venom patients. That is something that might be of interest, for example, for inhibitor studies, stuff like that. For example, as we saw with the black mamba, they still had some activity. Of course, in bite patients, the neurotoxicity is prevalent. But if a victim then survives the neurotoxic effects, it might be wise maybe for doctors to observe the patients a bit longer, also for effects like bleeding or onsetting necrosis, which might take a while to set on with these species. And then the limitations, of course, we only tested individual components or mimics of ECM. So the structure is not as complex as in our cells, but it gives us a good idea which components get cleaved. And that shows us, okay, here we have some very ECM active components or not. Then thank you to Associate Professor Dr. Jan Kuhl for being my supervisor, to Matthias Bittenbinder for being my supervisor in the lab, Professor Dr. Nicholas Casewell from the CSRI in Liverpool for venom samples, input, and all the other collaborators and venom collectors, which without them, this project would not have been possible, and to Dr. Wolfgang Wüster for photographs of the tested species here. Thank you for your attention. Please feel free to ask questions. I'm happy to answer them.
Yeah, thank you, Eric, for the talk. And as you said, any questions, then it's now your time to ask them directly or type them into the chat.
Hi, it's Mark Wilkinson, Liverpool. Can I ask questions?
Sure.
First of all, Mike, I totally agree with what you said at the start about biochemistry. Eric, at the moment I've been working. I've got some purified SVMPs from Echis ocellatus, and I've been studying the specificity.
Yeah.
What I thought was interesting from your result. Can you put your slides up again? Is that?
Yeah, sure. No worries.
So is that SEC result?
Sorry. There we go.
That's the one, yeah. So with the ocellatus, I've got the P3s and the P1s purified, and I find it very different. The P1s act on very few substrates, but your results show that the P1, that's that second peak, the smaller protein, is much more active than the P3. That's the first time I've seen that, and it makes me wonder if the P1s are responsible for the damage to the basement membrane, and the P3s are responsible for damage to blood to blood protein. So that's a really interesting result. And also the rhodostoma, that's a SVM P1 that seems to be very active as well. So can I ask if I can email you and get your assay methods, because I'd be really interested in using that assay on my purified. I've got some P2s from other Echis as well, and at the moment I'm just assaying them against small peptides to look for specificity, to look for which particular bonds they cleave. So, so I really enjoyed that. So yeah, can I email you and get the assay method from you?
Sure, you can email me. I can't give you the assay method yet because we have a paper due to be published hopefully soon, or sent to a journal hopefully soon. And once it's accepted, I'm happy to share the essay with you, or you might discuss with Dr. Jeroen Kuhl, who is, tightly collaborates with Professor Nick Casewell. Maybe we can work something out that way.
Yeah, okay, yeah. I didn't realize it was a novel method, so yeah, I can wait, that's fine. The other thing I was going to say, if you coupled SEC with nanofractionation, the nanofractionation uses reverse phase chromatography. And I know from experience that will kill the activity of metalloproteases.
Yeah, that's why we use SEC.
Yeah, for sure, yeah. So I tried, because the P3s are a complex mixture, I tried using reverse phase to separate the individual forms, and I tried including calcium and zinc with the reverse phase buffer, which has worked for proteases in the past, but it still killed the activity.
Yeah, I can. I don't want to give away too much due to the paper not being accepted yet. But I can tell you that we tested certain mobile phases that are frequently used with this assay, and basically incubated the venoms in them. And yeah, they completely nuked the activity. So even they
killed the activity.
The mobile phases are way too harsh for the large proteins, and they just, especially some of them, they are not like one protein, but because they're protein complexes, I think it just nukes them completely. So yeah.
I can't wait to see this paper. Thanks, Eric. I'll close there.
Thank you for the question. Okay. Anyone else?
If nobody else, then I would have a question to slide 7. So you showed that the different Botrops venoms are highly different depending of if they are from South Brazil or from the southeast.
Yeah.
From which location is your one that you shown?
Sadly, I don't know because it was not listed. That's why I showed both. But maybe I have to double check, but I don't know that we even have that in the database, because some of the venom samples are fairly old. So sadly I can't tell you which.
But do you have maybe an HPLC chromatogram that you can compare? It might be with the one that are shown in the literature, so that you might be more get a guess.
Yeah, we could do that. But as of now, I can't tell you. Sadly, the same with Callosolas marodostoma. I mean, the main like share of SVMPs is basically almost the same throughout the range, but the different subclasses have different amounts depending on the locality. So actually comparing these would be really nice with the literature to see from which they are, or at least narrow it down to a certain region. Yeah, that would be a good idea.
Especially with the both of this would be awesome with it if you get this with just with the 10% venom. That would be super effective.
Yeah, the boat drops is just first I was like, oh, did I not pipette my positive control correctly or something? But yeah. It's
sorry, Mike, Eric, there is a, because we recently did some work with Jararaca, so I had to look into what protease was there, and there's a nice proteomics paper that compares the content of the South and the Southeast Brazilian. I can't remember the authors, but Juan Calvet was one of them. So,
okay.
So it's fairly recent, and it really gives a nice, again, you know, the separate classifications of its. Well, we think it's P3 and P1 in that particular venom. There's quite a lot of serine protease as well, but I guess serine proteases are not active in your secret assay, are they?
I don't think so. We might, of course, we have to check that with the active fractions, which is in the works now. But the only thing that I can see them cleaving is maybe the gelatin, because it's like basically pre-digested collagen type 1, but this would be very minor because they usually have different targets. So
yeah. I'll send you that paper when if I can get your email address.
Okay, yeah, thank you.